Good morning, my name is Joana Cabral, and I will talk about macroscale oscillatory wave patterns that have been revealed with ultrafast fMRI. So, overall, for now, let's start with what is a standing wave. Standing waves are waves that are fixed in space and oscillate in time. They are characterized by the existence of nodes. Nodes are points of no motion, so they are points in the line that do not move in time while the standing wave is on. And then there are the antinodes of a standing wave, and these do oscillate in time. And these antinodes can have different polarities, which means that you can have anticorrelations between distinct antinodes, but essentially you can have correlations between antinodes with the same polarity. And this drives functional connectivity within all the antinodes with the same polarity. So standing waves are driven by resonance phenomena in bounded structures, and they can be predicted from structure using the eigenvectors of the Laplacian, as you've been seeing in other talks. And overall, they are quite universal. They can be observed in one dimension, like in strings and tubes, in two dimensions, like in the famous Chladni plates, or in membranes, or even in three dimensions, such as in structures or resonant chambers and rooms. As you can see, the activation, so the same structure can have different possible standing waves. And as you see in the middle, in the red and blue colors, all the points that are in the red areas, when that standing wave is activated, they will oscillate in phase, so they will drive a given functional network, right? So we can assume that different standing waves can drive within the same structure different functional networks, even if there is no clear anatomical boundary separating the different regions, right? So a different mode can create different structural boundaries where, for instance, the sand accumulates because it's the points of no motion, even if those boundaries do not actually exist physically. So brain activity has been for some time proposed to be the result of the superposition of standing waves. This has long been proposed using EEG and MEG activity, using neurofield theories. So there are different approaches. For instance, here I'm showing a paper where they reconstruct into spherical harmonics that are then expanded to the surface of the cortex, showing that the functional networks we observe may be the reflection of a standing wave. Then in 2016, for example, Selena Tassoy used instead the connectome, the graph Laplacian. So she used the connectome structure and applied the graph Laplacian to detect the eigenvectors of the graph Laplacian, and showed that these reveal features of functional networks like we see in the brain. And more recently, this was extended to consider, instead of the connectome, considering the surface mesh, that we also observe similar patterns of standing waves that can be used to reconstruct brain activity even more accurately. So overall, this to say that there are different approaches to put forward this hypothesis, but they converge in the sense that they propose that macroscopic brain activity is linked to standing waves or resonance phenomena. So overall, as you've seen, like there is this traditional approach to go from the structure, taking either the surface or the graph or the shape of the brain to compute the Laplacian eigenvectors and predict the functional networks, or from in another field, also in neuroimaging, is to take the time series of activity, apply some component analysis like principal component analysis, independent component analysis, gradients, leading eigenvector dynamics analysis. So there are different approaches that reveal functional networks as well. And the idea is to see how similar these are, the ones that are predicted from the structure and the ones that are extracted from the time series, to see if there is at least a qualitative similarity between the two that supports this hypothesis that the functional networks that we are detecting, the intrinsic connectivity networks or resting state networks, are indeed the expression of standing waves. Now, there's one thing that is usually not fully addressed, is that in standing waves, the signals oscillate in time with most specific phase relationships. So, which means that some voxels or some units in the system, so this is a continuum system, where some parts of the system will synchronize or oscillate in phase, whereas others will oscillate in antiphase with each other. So it's important to understand if the functional networks that we see really in brain activity, do they actually oscillate in time, to further validate this hypothesis. So to do so, I collaborated with a group at the Champalimaud Center for the Unknown in Portugal, where they have a 9.4 MRI scanner for rodents. And I asked them if with how fast we could get fMRI signal, because the fMRI data that I have usually have very low temporal resolution. So we only have one signal every around 1 second, and it's very hard to see at the instantaneous level how sustained or how oscillatory the activity is or how periodic it is. So we focused in a single slice. And by focusing in a single slice, we don't have to change the slice inside the scanner, so this really accelerates a lot. We also played a bit with the parameters in the scanner, and we reached acquisitions with a TR of 38 milliseconds, which means that in 10-minute scans, we have 16,000 frames per scan. And then we used 6 rats, and each rat was scanned twice in 3 different conditions. In the first condition, they were only sedated with medetomidine. Medetomidine is known to not affect the bold response, but also to drive some oscillations of unclear origin in the fMRI signal, which I was particularly interested. And then we added 1% isoflurane, as is typically done to kill those higher frequency oscillations that are seen in fMRI and to approximate the human resting state activity. And then we added isoflurane at 3% concentration. And in that case, it is known that usually the fMRI signal is really vanished, and we lose all types of functional networks that can be detected. So in total, we have 12 scans per condition, so 36 scans, and we also did 2 postmortem scans to serve as baseline. And the first thing we did was to analyze the power spectrum, right, of the signals. In each voxel, we calculate the power spectrum, and we see which voxels have power in which frequency range, right? So we divided here until in bins of 0.5 hertz, and we find that there are oscillations peaking in the cortex up to 0.3 hertz, and also found in the striatum, although slightly, it's a bit slower frequencies. These oscillations were found to decrease with the addition of isoflurane anesthesia, as predicted. So as you can see in the second row, we have slightly less oscillations, particularly there at 0.2 hertz. So we find that isoflurane clearly acts on the power of those oscillations, and if we further increase the concentration of isoflurane, we lose most of the power above 0.05 hertz. Another thing that is, and we compare this is relative to the post-mortem scans. So how much power is, so the voxels are colored in according to how much power they have above the post-mortem scans. And we also did statistics. One thing that is interesting here, I highlight with a shade the typical range of frequencies considered in resting state studies. Here we wanted to explore the whole range of frequencies, so we analyzed up to the Nyquist frequency, which was close to 13 hertz, but we found that above 0.4 hertz, only power associated with breathing and heartbeat was detected. So the range of frequencies that really showed the most significant differences across the 3 conditions was there between 0.2 and 0.25 hertz. So what we did was to take a scan, like a scan of a sedated rat, and we filter in that frequency range. And I will show you here what we see. So if we do filter the signals between 0.2 and 0.25 hertz, we see signals that exhibit sustained periodicity For instance, if you focus there on the lateral sides, you see that consistently, so it turns blue, which means there's a negative signal, then it turns red, means it's positive with respect to the mean signal. And then it's again red, and then it's again blue, so there's a sustained periodicity, and we find that these oscillations can be correlated across distant areas of the brain. Here, the contralateral areas exhibit high correlations, also with the middle part of the brain. So one important thing to see from this is that first, the oscillations we detect are not fully synchronized, so everything is not just going up and down at 0.2 Hertz, they are organized in space, and also they are organized in time in the sense that we don't always see exactly the same pattern of activity over time. So there are some switches between the patterns that we see. So here, for example, the middle line is in antiphase with the lateral areas. So now we did an analysis. So now I will show you here the consistency across different animals, right? So we see in the sedated condition, I'm showing here 3 rats. Here I put a bit broader range of frequencies, so between 0.15 and 0.25, but we clearly see that these patterns of activity that we see switching between positive and negative values with respect to the mean is consistent across the 3 rats, right? Now, if we add a bit of isoflurane, we see that these patterns are changed. We see perhaps more global signals in the cortex. They are more noisy, apparently. So we adjusted the color bars to take into account the reduced signal with isoflurane, but still the signals are a bit more less organized. And when we add isoflurane at 3%, as you can see, the macroscopic organization is lost. It's harder to detect any type of structure in the signals, and except from some very slow, like activation, like you saw now this plot in the middle, the rat number 2, that can show like everything turns blue, then everything turns right, but it's a very, very subtle global mode. So what we did was to analyze the components of the signal. So those oscillations, so we have the broadband signal, we filter up to 0.5 Hertz because this was the range where we found most activity in the cortex, and then we apply principal component analysis, but we do it at a group level. So for each scan, we compute a covariance matrix, and then we average the covariance matrices. We take the leading eigenvectors, sorry, not the leading, all the eigenvectors that have an eigenvalue above the postmortem baseline. Okay, so we do the same covariance matrix for the postmortem scans, and we take the leading eigenvalue, and we sort them in this direction. And we find, and using those patterns, we can plot the temporal signature associated with each pattern. So we can see that the signals, when we look at the temporal signature, we find that the temporal signature oscillates in time, which means that the patterns switch between positive and negative representations of themselves, the components, in a periodic way. And this leads to a peak in the power spectrum. So there are these features that are really important to capture, that these principal components exhibit nodes and antinodes in the wave patterns. They exhibit some modes, not all, exhibit antiphase relationships in the sense that some antinodes are in phase and in antiphase with the others. And also that they oscillate in time. So here, for example, I'm showing mode number 2 multiplied by its temporal signature. So as you see, it oscillates, it fluctuates between positive and negative representations of the mode, right? Now, if we add these modes together. Like when we decompose a system into 10 components, then we can reconstruct it again, since they are, in theory, the eigenvectors form an orthogonal basis, which means that they can be added up linearly. And in this sense, it is said that it obeys the wave superposition principle. And what we find is that this reconstruction, although so each single mode is just a standing wave, when we add them all together, we see visually like some traveling waves that can be generated. They can go in different directions. Here they seem to be going up, but then at some point they switch and seem to be going down. So you see like this superposition of standing waves can generate really interesting phenomena at the global level, at the systemic level. And also, of course, there's a high dimensionality reduction, since the scan on the left has like 24 million bytes, whereas on the right it is just described from 31k bytes. So what here we show is that when we add isoflurane, it reduces the number of components detected. So before we had 10, here only 6 passed the threshold of the postmortem. We find that oscillations are more damped and less periodic. And then medetomidine and isoflurane is known to approximate the human resting state activity. So these oscillations are less clear, but they are still there. So the modes are still phenomenologically similar to what we observed in medetomidine. Now, when we add isoflurane at 3%, what we find is that there's only a single mode that passes the threshold. This mode is global, so there are no anticorrelations, and there's no sustained periodicity, so there are just kind of aperiodic global fluctuations lasting over 20 seconds. So we try to compare this across conditions. Of course, since the modes are different, what we compare here is the resonance Q factor of each of the modes. And the resonance Q factor gives us an intuition of how long, like whenever a wave is activated, like how many sustained cycles it activates before it vanishes. And we find that under medetomidine, the resonance Q factor is higher. It is with the addition of isoflurane and the peak frequency as well. With the addition of isoflurane, the oscillations are more damped, which means that they decay faster. And with isoflurane 2%, there's really the peak frequency is super slow, and there's really no reverberation, no periodic reactivation of the same mode. So this reminds us of to understand what are the regimes of harmonic motion. So there are like equations that describe in general different types of response that a system with standing waves can exhibit. And usually, so in theory, you could have a system that is always, forever and ever oscillating between positive and negative values with constant amplitude is said to be in a limit cycle. And here it's represented in gray. But this in reality doesn't occur in nature. Usually the oscillations are damped. They eventually decay over time, even if it takes years, right? So these are called damped oscillators. And in the damped regime, you can have two scenarios. Either it's underdamped, which means that when it's activated the wave, like imagine when you play a drum and then it does until it vanishes, or it can be overdamped, which means that you play it and then it just boom, decays now back to baseline. So both damped regimes eventually decay to a fixed point equilibrium, but in the underdamped case, it will resonate at the natural frequency of the oscillator. And which is interesting here is if you consider a damped oscillator and just an underdamped oscillator and perturbed it with white noise, you'll see that although white noise has all the possible frequencies, the power spectrum of the time series generated really peaks at the natural frequency. So these are noise-induced oscillations, so kind of stochastic resonance of a system. This system would be at zero, not active if it's not perturbed. If you perturb it with white noise, it will eventually resonate at its natural frequency. Now, if the system is overdamped, we don't see anymore this peak. So clearly, the spectrum is more broad, but still peaks at lower frequencies. So this approximates, we find that the temporal signature of an underdamped oscillator approximates what we observe in medetomidine in the response of the standing waves. And with high damping, we find that it approximates what we see with isoflurane anesthesia. So we kind of try to model what would be the activity generated by the stochastic resonance of the standing wave. So we take the standing waves that we detect empirically, right, from the data. So we have these 10 modes, and we make them oscillate, right, generating in silico, using the Stewart-Landau equation, the response of an underdamped oscillator perturbed with white noise. And so with this, we can create brain activity. So we can superpose all these modes. We make them oscillate, we superpose, and we generate a pattern of brain activity. And as we show here, like different snapshots in panel D, we see that we can generate a multitude of possible patterns just by the superposition of these different waves. And essentially what we wanted to show is then if we compute the correlation matrix of the generated signals, this will reveal long-range functional connections or long-range correlations, let's say, that can be understood as functional connections. So it's a possible mechanism to explain why we observe this spontaneous functional connectivity at rest. This is to show as well that the results that we found in the single slice can be extended at a whole brain level. So of course they are not specific to that specific brain slice. So these modes really seem to be propagating across the entire cortex. Here the temporal resolution was lower because we have to change the slice. And overall, like here, I render here the modes that we detected at the whole brain level, so positive and negative representation. And the idea here is to show that even if you can predict the modes from the structure alone, like you can never really address the temporal signature because this will depend also on other properties such as the resistivity of the medium, right, through which the waves propagate. So anything that modulates this resistivity can change the temporal signature and even the spatial features of the standing waves in the same system. So overall, this is like a cartoon representation of how the activity that we observe is actually the superposition of multiple modes. So in a way, it's like a melody or like an orchestra that is composed by the sum of different components. Those individual components can be completely reversible in the sense that if you flip them around, you don't see any difference because they are oscillatory. But when you superpose, you generate reversible or irreversible and turbulent dynamics. Overall, I want to stress, so first to say that our results align with this emerging hypothesis that functional networks are driven or are the expression of standing waves resonating transiently in the brain. And it's very important to understand the mechanisms modulating the activation of these standing waves over time, in order to address, to rebalance, let's say, the functional networks that are disrupted in mental disorders. So now I think this is the way to go to really understand what is going on and how we can modulate these modes with precision. Thank you very much for your attention. This work was done in collaboration with the Shemesh Lab at the Champalimaud Center for the Unknown, but of course I want to thank all my colleagues at the University of Minho and the extensive networks of collaborators that I have that have discussed these ideas. Also my funding agency, the La Caixa Foundation. Thank you very much.